A common approach to studying eukaryotic cells is diminishing production of a protein. To accomplish this, we have two unique methods, RNAi or CRISPR. Both techniques have some similarities but contrasts as well. As a scientist, if I wanted to diminish the function of a specific gene, let's say in yeast, for which there are no known strain variations or mutations, I'd rely on CRISPR. Let's take a look into why. RNAi was originally found to combat foreign gene expression from viruses. The tool RNAi uses to accomplish this is by using double-stranded RNA molecules and a protein called dicer. Depending on the origin of double-stranded RNA, the resulting RNA molecules produced can be classified as siRNAs or miRNA. Small interfering RNAs target the genes that made them and originate from mRNA, transposons, or viruses, while microRNAs are transcribed from a distinct gene and will only attack genes other than its own. If the double-stranded RNA does not originate from a virus, then it can be transcribed from inverted repeats in DNA sequence, forming a hairpin. Once fully processed, RNAi molecules combine with proteins to create RISC. RISC can then go on to either inhibit translation of a target mRNA or cut target mRNA to allow an enzyme like RAT1 to degrade it. As amazing as this sounds, in the lab, RNAi has some limitations. For example, interference is usually temporary. Once the RNAi molecules are depleted, the interference ends. Also, it's possible that RNAi could halt production of other proteins as well, and if it does, then you can't be sure if the results you see are because you confirm targeting of a specific gene's expression or that you just have similar mRNAs that can interact with the RNAi. That is why I favor CRISPR. CRISPR RNA was discovered as a natural defense system against bacteriophage DNA invasion in prokaryotes. Scientists learned to adapt this defense system to directly modify DNA genetic sequences in eukaryotic cells. CRISPR works by activating a class of Cas proteins like Cas9 in combination with a guide RNA to target DNA. The Cas protein then cuts away the target section of DNA, causing a random mutation to fill in the cut section. However, if you need to be more precise, you can use the CRISPR-Cas9 system to also introduce a pre-selected DNA template to ensure the gene is silenced, or replace mutated DNA with a healthy DNA and vice versa. Delivery of the system can be completed by a viral vector, by introducing DNA needed to code Cas9 in the cell as well as delivering the guide RNA. As ideal as the system is for my study, it also has some drawbacks. For example, if you're unsure about the exact function of a particular DNA sequence, then you may wind up inducing undesirable effects, and in medical therapies, this kind of mistake can be deadly and irreversible. However, CRISPR is known to be much more accurate than RNAi, reducing the chance of off-target effects. In a laboratory setting working with a model organism like yeast, for example, even if I get an off-target effect after observing a few generations with the modification, then I can be more confident in associating the effect with manipulation of the gene sequence I edited, allowing me to further refine the technique.